Welcome to RAADcast, where science and soul converge in shaping the future of human potential. I'm your host, Dr. Melissa Grill-Peterson, and together we're stepping beyond limits to explore super longevity. Produced by the Coalition for Radical Life Extension, the visionaries behind RAADfest, RAADcast is more than a podcast. It's a movement for the bold, the curious, and those who refuse to settle for anything less than the extraordinary. Together, we'll uncover the cutting-edge science, unknown strategies, and transformative practices, paving the way for a future of limitless potential. If you're ready to think bigger, dream bolder, and live longer, you're in the right place. This is RAADcast. Now let's get ready to explore the future of life itself. Hi, everyone, and welcome to RAADcast, where we bring you the latest breakthroughs in super longevity, health optimization, and the science of reverse aging. I'm your host, Dr. Melissa Grill-Peterson, and today we are diving deep into one of the most critical mechanisms of aging, our telomeres. And who better to teach us about this and have this conversation than world-renowned guest, Dr. Bill Andrews. He is a molecular biologist, longevity researcher, and the founder and CEO of Sierra Sciences. His groundbreaking work has revolutionized our understanding of aging by focusing on telomere lengthening, a process that could hold the key to extending human lifespan and health span. Thousands of scientific studies have shown that our telomeres shorten, we age, and our health declines. But what if we could stop or even reverse this process? Dr. Bill Andrews, you're the one who can shine some light on this. So we are honored to have you on the show today. Welcome.
Well, glad to be here.
I always love getting time with you, and I really appreciate it. I know you're having a snowstorm out there right now, and so I want you to stay warm as you take us into the journey as we really kind of start with the discovery of telomeres and aging, because telomeres have been studied for decades, but your work has really, I would say, truly advanced the field. Could you walk us through how you became involved in telomere research and why it's the potential key to longevity?
Well, it has to go back before even telomeres. The question's always been in my mind. I've been interested in aging my entire life, but why do we age when our cells, we're made up of cells. We're not like trucks sitting in a field that are suffering from wear and tear. Yes, we do have wear and tear, but we have cells that can divide to replace those damaged cells. So that's been something that even in high school, when I was doing anti-aging clubs, that was the discussion that we talked about the most.
Did you just say you did an anti-aging club in high school?
Yes. Oh, yeah.
Oh my gosh, we have to talk about that too. Okay, keep going. So you're in an anti-aging club. You're already thinking at a young age about this. I didn't mean to stop your flow, but that is fantastic. Keep going.
No, actually, the story, if you ever watched the Immortalist movie, the documentary that almost won an Oscar, that stars me and Aubrey de Grey,
Yes.
it clearly shows that my father was the one that got me interested in aging when I was only 10 years old. I mean, like teachers would send me home with notes on my shirt, pins in my shirt, and my parents would read it and said, "Your son's very interested in science and medicine. You should nurture that." So my father, one day when I was 10 years old, just came up to me and said, "Since you're so interested in science and medicine, when you grow up, you should become a doctor and cure aging." He used the term cure aging. And he
Wow.
also said, "I don't know why nobody's done this yet." So he had this perception that aging was just a disease and stuff like that. So I've always thought so too. But yeah, no, so high school and college, I was organizing anti-aging clubs and stuff like that. And this is like 1960s, 1970s, things like that. So it's been a long obsession. But the big problem was okay, so the number one cause of aging is wear and tear. That's
Yeah.
no question about that. But why does wear and tear cause us a problem when we have the ability to have other cells to divide to replace those damaged cells, damaged from wear and tear?
Yeah.
And so that's always been a mystery. And then back in 1961, Leonard Hayflick announced that he had discovered that human cells only have the ability to divide a certain number of times. So there's a so something's going on that's making it so that our cells can only divide like at the most 100 times from being a conceive. Well, once you're born, your cells can
Mhm.
only divide about 50 more times.
Okay.
And after that, they go into a phase called senescence and they lose the ability to function. And suddenly we can't repair our damaged tissues, our organs, and things like that because there's no cells to divide to replace the damaged cells. So I remember we would talk about how could a human cell, know how many times it has divided and how
Yeah.
many more times it can divide. And I remember thinking there's something in like keeping score in like when you're playing pool, like with, you know, a pool table. Um, you hit a ball into the pocket and you take your cue stick and you reach up onto the wall and you slide this wooden thing from one side to another to keep score. And we
Mhm.
used to talk about things like that. Was there something like that in a cell? Because how could a cell have such a mechanism? We knew enough about the biochemistry of stuff like that that we knew it can't be just like a calculator inside the cell. Cells don't have brains. They can't think. There's some scoring mechanism that's occurring. But the best one that we came up with was ride tickets at an amusement park. There's something like ride tickets at an amusement park. And so when a cell divides, somehow automatically a ticket is removed. And then a cell divides again and another ticket's removed. So you only have a certain number of tickets, and you can, scientists can, if they knew what the tickets were, could count the number of tickets and figure out how many more times a cell has to divide. So question is, what, where, and where could this possibly be? And so, you know, I don't know if you're interviewing Mike Rose, but I hope you are. But
Mhm.
one of the things that happened was back in 1993, um, I was attending a conference in Lake Tahoe on aging. And Mike Rose and I decided to start a company together to try to figure out the aging process and to solve it. And so we stayed up all night planning it and things like that. What we were going to do we all came up with the name was going to be called, oh, what was it called? The Methuselah Project, or Project Methuselah was the name of
Nice.
the company.
Nice.
But then the very next morning, a guy named Calvin Harley. Oh, by the way, my father was at this conference too. He and I would go to these conferences together because he was so interested in anti-aging.
Yeah.
The very next morning, a guy named Calvin Harley stood on stage and talked about telomere shortening. I
Mhm.
knew what telomeres were. Everybody knows what telomeres are if they have even a class in biology. But he said that they shorten as you age when cells divide. And I'm thinking to myself, oh my God, here's the ride tickets. This is it. And so before he could even get off the stage, I asked him, has anybody figured out how to prevent the telomere shortening?
Mhm.
And he says, we have been working on it for years, collaborating with labs all over the world, and we have had no success whatsoever. And I just said, hey, bring me on board. I'll figure it out in 3 months. Okay. And I mean, I had already a big history of successes in biotech, all the way from human growth hormone and erythropoietin and tissue plasminogen activator and beta seron, just on and on. So he ended up pretty much offering me the job on the spot. And 3 months and 17 days later, we discovered why it is that uh, human reproductive cells, essentially primordial germ cells,
Mhm.
don't have telomere shortening. And that was because they have this enzyme called human telomerase.
Mhm.
And so that was the launch. But going back to that conference, um, the very next, right after that conference, I went up to Mike Rose and I said, hey, I can't do this company anymore because I just agreed to go work with at Geron Corporation with Cal Harley to figure out the telomere biology problem. But I brought my father with me when I told Mike Rose this, and I said, but my father would like to take over my role as president of the company. And to my, I mean, I was not too happy about what happened next. Is Mike Rose got really excited about it. Oh, great, great. My father had been very successful at starting other companies and stuff. And he was a well-known television producer and had lots of success doing that. And so he was all excited about that. And so my father and Mike Rose went ahead and started Project Methuselah. And I went to Geron Corporation and worked on telomeres and telomerase. But that's pretty much how it started for me. There's no way, I was not the person that came up with the idea that telomere shortening solving the telomere shortening problem was the solution to aging. Calvin Harley published a paper along with Richard Alsop on the telomere theory of aging. And of course, I didn't know about that paper until after I heard his presentation. But that's just took over my entire life after that, because I was trying to solve the problem of why can't we replace damaged cells from wear and tear, and suddenly solving the telomere shortening problem became the solution for that. And almost all the hallmarks of aging are related to wear and tear. So I believe there's a good chance that solving the telomere shortening problem will solve all the hallmarks of aging, or most of them. And plus, I think given what we do know about telomere shortening, there's no way we could ever successfully cure aging unless we, in addition to whatever else we do, we solve the telomere shortening problem too. So that's why my focus is on that. But I actually don't consider myself a telomere biologist. I consider myself an anti-aging scientist who's right now focusing on trying to solve the telomere shortening problem. But I'm also working on other things too.
The longevity revolution is about more than just searching online for a few hacks, or any one of us moving our biomarkers in the right direction. It's about coming together to learn, share, to connect, and create the unlimited future we want. That's why you need to be at RAADfest 2025 in Las Vegas from July 10th to the 13th. RAADfest, the revolution against aging and death, is proud to be partnering for the first time with Dr. Aubrey de Grey and Longevity Escape Velocity Foundation to form the single most comprehensive, advanced, informative, and inspiring event on age reversal for a general audience. Meet the people you want to make forever with. RAADfest 2025. Take advantage of lower advanced pricing now at raadfest.com.
I appreciate it. Well, I want to hear about the other things, but I do want to, first of all, thank you for the foundation. And I love your journey. I love knowing how this started. It's interesting, when James and we've had different conversations, and for James, his journey started when he was young with his grandmother, right? I talked about how when I was 10 years old, and it's interesting that you said you were 10. I remember just having, and I don't even know why I had this thought, but I come from primarily Italian heritage, and it was a big family gathering, and I was just looking around, and my family's very robust, and they're filled with life, and they're smoking and joking, and they're telling stories, and they're just having a grand old time. And I just looked around at all of them. They were beautiful, like just gorgeous, and like my great aunts. I'm like, gosh, I'm gonna live a really long time because I have great genes. That's what I said when I was 10. And I'm like, where did that even come from? But there's something really interesting how many of us that love this field seems as though there was some kind of spark, some kind of connection when we were in our early childhood years. And I just found that really interesting. I love the role that your father played in really supporting your journey along the way. And of course, we all get to benefit from that. And so you went to work within 3 months and 17 days, you found kind of that next piece, which was all around telomerase. So talk to us about like, and it was in those primordial reproductive cells. So it's inactive unless we're in that reproductive state. So how has your work looked at really activating? When should we activate? Do we activate? How do we activate more telomerase to potentially really, solve this telomere shortening issue like you've discovered?
Well, the first approach that we did was to first isolate the promoter that regulates the gene for telomerase and try to figure out what's repressing it, what's shutting it off.
Mm.
And so we worked for several years on that. And we found what we consider to be a lot of repressors, different repressors, or candidates for repressors. But when we inhibit them, the gene still didn't turn on. And so we did more research, and we concluded that there was actually multiple repressors, multiple types of repressors binding to multiple repressor sites in the DNA motor. And that the only way we were going to turn the gene on was to inhibit all of them at the same time. And so we'd have to know that we knew what all the repressors were to do that. And so far we hadn't succeeded. So we decided to take a different approach at that point, and was more working blindly. And that was we developed an assay, high throughput robotic assay, that would measure whether or not a cell, human cell grown in a petri dish, or at least a microtiter dish, was producing telomeres. And so we could test 4,000 different cell lines a day to see if any of them were producing telomerase. So we set up this assay, and we started doing our screening. We started testing chemicals at random just to see if adding the chemical to the cells would turn the telomerase gene on. Now, a lot of things we already knew would do that, like histone deacetylase inhibitors and things like that. But the problem with things like histone deacetylase inhibitors is that they turn on 80% of the genes in the cell. And so, that causes all kinds of problems. And so we needed to find something that would be specific towards just the telomerase gene. So we ended up quickly finding, like, I want to say 60,000 different chemicals. No, we tested 60,000 chemicals, and we found about 100 that actually did turn on the telomerase gene. Okay. Then we started doing medicinal chemistry, where we started redesigning these chemicals to make them stronger and safer. Because the chemicals, when we were doing random screening, they would oftentimes have side effects and things like that. So we tested another, like, maybe 300,000 different chemicals, mostly which we designed. And we ended up finding close to 900 different chemicals that would turn on the telomerase gene. But none of them turned it on to the levels that we needed. The highest level we've gotten so far is we can get the telomerase gene to turn on to 16% of what is needed right now to stop telomere shortening. So turning it on a little bit, it will at least slow down the rate of telomere shortening, because shortening and lengthening is like a tug of war.
Yeah.
Adding people to the side of this lengthening, but they're still losing the tug of war. So one of them was considered very safe, has been through clinical studies, and is now marketed by a few companies and stuff like that. But that's where we're going. We still have not figured out really what the repressors are. There's been lots of papers published saying that they discovered the telomerase repressor, and in all cases we've tested them and found that it's not true. We can't reproduce their data. But when we talk to them, they say they can't reproduce it either, but at least they got
but they had that one time.
Yeah, well, it's like the publisher perish.
Yeah.
It's just it gets people to publish papers just so they can say they have a lot, because it helps them get grants. But too many times papers get published that nobody can reproduce. But yeah, so that's where we are right now. The number one goal right now is to find the funding to continue that research, which costs us essentially $2 million a month.
Wow. I mean, that's a big price tag. And it's a moonshot, meaning you're doing big work that fundamentally could do exactly what your dad told you should set out to do, right? Which is to cure aging.
Let me say, the moonshot comment. If I could quote James Stroll.
Yeah.
We're already there.
Yeah.
So that's the way to think about it.
Are you ready to redefine aging and unlock your full potential as a longevity leader? At the Human Longevity Institute, our world-class certification programs train doctors, coaches, and wellness professionals how to deliver cutting-edge integrative longevity health solutions so people can live better today for longer, healthier, and more vibrant tomorrows. If you're ready to join the longevity revolution, Visit humanlongevityinstitute.com to enroll and become a practitioner today. I love that. Yes, it's not like this is some crazy far off idea, meaning you are a big thinker. So that's what I was meaning by that. And thank you. Because I agree. I believe we are at a time that it is all here. And the funding, like keeping it all going. So there's kind of this balance of, there's still more to go. So you've made the foundational discoveries. There is a compound that is available that you said it's helping some. It slows. It doesn't stop fully, but it does slow. So it will activate that telomerase. So two things. First, are you able to tell us what that compound is? Or is that something is it a pharmaceutical? Is it a nutraceutical? Is it something that we can easily access? What
Well,
is it?
I don't market, I don't market product, but I'll just say we have pharmaceuticals and nutraceuticals.
Okay.
Our most potent nutraceuticals are going to be launched in early April by a company called Touchtone Essentials.
Okay.
And our most potent pharmaceutical has only been approved for skincare so far, and it's skin cream. And it's marketed by a company called One Truth 818.
Okay.
They've taken it through clinical studies and stuff like that, and it's safe.
Great. So it's exciting to know there's some things coming to market. So question, and I know I've kind of bent your ear on this before. Let's talk about lifestyle. So what are things that we as individuals right now should be thinking about that we could do either more of or less of that would potentially support, can we activate telomerase activity? Can we help to slow down the shortening again through some of our lifestyle choices, habits, environmental design? What's your thoughts on that?
Well, there'll be several phases of answering that question. But first, let me say there is absolutely nothing that you can do in lifestyle right now that will induce telomerase expression. There's lots of studies reporting that you can, for instance, like hyperbaric chambers, they're reporting that you can induce telomerase and lengthen telomeres. There's papers saying that exercise does. But they're all doing the assays incorrectly, or they're misinterpreting their own results. And I'm the inventor of most of the assays that are the tests that are done to test for activity. If anybody ever sees a study reporting that they have a therapy or drug or anything that produces telomerase, they have to look for what's called a telomerase trap ladder, which is the only assay that really means anything. And if you don't see the trap ladder in the study, it means they don't really have its activity, because that's the first thing anybody would do. And a lot of times they try to do it and they don't get a trap ladder, so they resort to other methods, and which are wrong, and which don't really work correctly, or they're not reproducible, but they publish them. Okay, the other way they do it is to measure telomere lengths. And again, the only really good way to really know that your telomeres are getting longer is to do what's called TRF analysis. And if they don't publish a TRF analysis result, that means their telomeres aren't really getting longer, because they are, again, resorting to other techniques that give false positives a lot. And again, I'm the inventor of most of these. I know from hands-on experience that there's
Right. So let me ask you a question. I'm curious. Is that so like True Diagnostics, for example, in their biological age testing, they do look at telomere attrition. So is the way that they're reporting and giving data on telomere length, is it to the appropriate standards? Is it something that we can have clinical relevance with, or is it still not? Are you familiar? I don't know if you're familiar with
True
their.
Diagnostics is one of my favorite companies on the planet.
Yeah, I love Ryan. The whole team's
Ryan
great.
Smith, yeah. I mean, he's a, but one of the reasons why I like True Diagnostics is that they don't rely just on telomere.
Right.
They put a multiple number of different biomarkers of aging and stuff. And so I think that his data that Ryan has shown at conferences and stuff like that correlates best with aging than anything I've ever seen. Now, I consider telomere length measurement to be too difficult to do to really get consistent results and stuff like that. So even though I believe that telomere length plays a role in aging, I believe that maybe other things are a better measurement of telomere length, and that includes DNA methylation and IgG glycosylation,
Yeah.
which I think those change as a result of telomere lengths. But your question was, what can people do now? And I've only answered the first one, is that
Yeah.
the most important thing is to decrease inflammation, decrease anything that causes wear and tear. Now, normal operations of tissues and organs, just normal things you can't slow down. So normal operations of what are called the frontline cells in your organs cause them to die off from wear and tear on the average about once a year.
Okay.
You can't reduce that, but you can accelerate that by all kinds of terrible things like smoking, and it includes increasing oxidative stress and increasing inflammation, which will cause the wear and tear to speed up. So you can reduce those and get it back down to the basal level like once per year. Now, when you lose frontline cells on your organs on average of once a year, that does mathematically put you at a maximum lifespan of 125 years when you calculate it mathematically. And that's because, the wear and tear of losing the cells once a year, and your cells only have the ability to divide 50 times after you're born. That says that, you're going to not make it past 125. And nobody makes it that far because of poor lifestyle choices and poor genetics that all accelerate that process. So decreasing inflammation, I highly recommend the ALCAT test, ALCAT, which is something I do once a year, because everybody's different. Different foods cause inflammation in different people in different ways. ALCAT is a test that allows you to be able to test whether or not a certain type of food will cause inflammation in you. And if it does, quit eating that food. And it changes every year. And then again, everybody's different. So I get tested every year, and I make certain that I don't eat the foods that are causing a lot of inflammation in me. And I get my inflammation markers measured every 3 months, and they're always undetectable. I just make certain that, even though I run every single day, a lot of people say running is something, I run and exercise every day. That says that causes inflammation, but not if you do it right. Keep it fun. Quit doing it if it's not being fun. It actually decreases inflammation. But so that's the thing, is like, exercise correctly, eat correctly, psychologically maintain a healthy psychological status. Those are all the kind of things that you can do to slow down the aging process.
So I'm very curious. Thank you for that, by the way. So you just shared that mathematically, our lifespan is 125 mathematically. And of course, a lot of the work is to say, can we lengthen that? Can we go beyond that? And as you said earlier in our conversation, you, along with Aubrey de Grey, were in the movie The Immortalist. So there's science and there's personal philosophy. Share a little bit of both with us about what you believe really is possible in going beyond 125. How soon do you think that could be with what you have access to, with what you see in the literature? And where do you hope personally to be able to go, since this has been a lifelong work for you? You're in movies called The Immortalist. So I want to hear a little science, a little philosophy of what do you really think is possible in extending beyond 125? How far can we go, and when?
Well, I think the best way to get close to 125 is listen to everything Aubrey de Grey is saying. I mean, he's doing everything that I'm not. I mean, not because I disagree with him, but it's like, why do what he's doing when he's doing it?
Right.
It's like, and I look at Aubrey de Grey as a person who's trying to cure my aging. So I'm supporting Aubrey in every way I can. And the same is true you know, Liz Parrish,
Yeah.
Sandra Kaufman, Greg Fay, Mike West, Mike Rose. These people are the people that are really doing the best there is to really solve the aging process. But as I said, no matter what anybody else does, the exception would be Liz Parrish because she is working on lengthening telomeres too.
Mhm.
Uh, no matter what else anybody does besides what Liz Parrish and I are doing, we'll never ever exceed 125 unless we also solve the telomere shortening problem. And
Mm.
incidentally, there is one reported case of somebody living to be 122. That was a woman named Jean Calment. And
Mhm.
he smoked like a chimney and things like that. So there was a lot of weird things that kind of didn't make sense. But now there's a lot of reports coming out that I've looked into, and I think they're believable. And that's that Jean Calment's daughter took over her life after Jean Calment really died,
Mm.
keep inheritance taxes low or something like that. It's and apparently there was some, Jean Calment was getting some money from inheritance from some different case. And so her daughter pretended to be her. And it was actually her daughter that died, not at 122, but when you added up the two together, it was like 122. So
Interesting.
I don't think anybody's actually lived to be 122. And Natalie Cole, I don't know if you're interviewing her, but she's somebody. There was a person, really the number one speaker at conferences that I used to really enjoy, a guy named Stephen Coles, who would
Mhm.
talk about the super centenarians and who they
Yep.
are and how they're doing and what stuff like that. And then he passed away from cancer, biggest upset of all time. And Natalie has taken over his role.
Mhm.
And so, probably to get a more accurate about things about what's going on with Jean Calment's real story, Natalie would probably be the best person to address that. But I think there's a good chance that actually she didn't really live to be 122.
Yeah. So for right now, well, personally, once, especially with Liz's work, and I just want to give a little plug right now to RAADfest, to our listeners, if you want to get to listen and learn and connect with these amazing minds and researchers at the forefront of the field, Aubrey, Bill, Sandra, Liz, they will all be at our 10-year anniversary this summer in Las Vegas. So go ahead and check the show notes below. You'll want to make sure you grab those tickets. And then the following year, there's going to be a big longevity march, aging march on Washington. So there's just a lot of amazing things happening with the coalition that's, really people coming together, experts, enthusiasts alike to say, all right, like there's got to be a way. We know more now than ever. And I do love Aubrey's his work, and I love what we can appreciate about closing the gap between health span, lifespan, and longevity escape velocity coming in there. Because every year we can kind of slow that down and, in essence, gain youth biologically, even if we chronologically progress forward. We are creating greater states of health and health span and function in the system that will keep us in a more vital state while the science catches up. And so I'm curious, if you had to make a guess, and we're not going to hold you to this, Bill, I promise, but where do you think, especially with Liz's work, how close do you think we are to actually kind of solving this telomere issue? Because if you, like you said, hey, we're not going to get past 125 if we don't also get to the bottom of this telomere issue. Like if we really don't solve it, like we can do all these other great things, take all the supplements, do all the fun stuff, but we're not going to really pass that threshold. Aubrey would tell us, that's going to be possible. And of course, he's been saying that for so very long, right? The first person to live to 150, would he started to tell us that about what, 15, 20 years ago? So this idea that we will pass that, kind of break through that sound barrier, how close do you think we are to that actually happening?
I think we could have a cure for aging in 1 to 3 years. Okay, I mean that's way ahead of what everybody else is saying, but
Yeah.
I think Aubrey would agree with what I'm about to say because he often puts it later. The number one obstacle to curing the aging process is sufficient funding. Okay, and that's, I mean, boy, put $200 million into Aubrey's hands or my hands or Liz Parrish's hands or it's Sandra Kaufman, Greg Faith. I mean, all of us. Uh, uh, it wouldn't take long because we know the science. The science is actually pretty straightforward. We just don't have the amount of funding that it takes to actually get this work done. And, you know, yeah, we all have no problems finding investors, but
Right.
investors are all interested in quick financial returns on investments. And then you start succeeding at doing things that give them quick return on investments, then they want to sell your companies.
Right.
And then you lose your intellectual property and you can't, non-compete contracts prevent you from continuing your work. And this is one of the obstacles that we all face. And so what needs to be done is we need to find a source of funding that doesn't restrict our science, doesn't change our focus. And that's really the big obstacle right now. But I think that if we had sufficient funding, I am a scientific advisor for Liz Parrish's company. I know a lot about the stuff she wants to do, but she can't do because of lack of funding, and not, or let's say, not insufficient funding.
Right.
And I want to create a whole department in my company. I've got 10,000 square feet of lab space right on the wall behind me, on the other side of the wall behind me, and I just want to create a whole division just focused on getting all the stuff that Liz Parrish wants to get done.
Yeah.
Using gene therapy. But the biggest problem with gene therapy right now is it's not safe to go to high doses. And
Right.
So, are the doses that are being used now sufficient? And I score things by what I call the Betty White test. I mean, somebody's got to go from looking and feeling and behaving 85 to looking and behaving and feeling like they're 25. And nobody's ever passed that test yet. So,
Yeah.
but I think I don't know if lengthening telomeres is going to do it all by itself. I mean,
Right.
there's no
Yeah. And I think that's, oops, sorry, go ahead.
we just have to get the funding to be able to do all the things and they all together to get us past 125.
And I think that's the opportunity. That's also the question, and it's also the cost, because there's the cost to you all in research to want to, R&D. Then what will that ultimately look like to the end user, right? How do we democratize these elements? So it's kind of good, better, best, freemium to premium. How do we, starting like knowing what we're working on, but then kind of backcasting to say, how do we do this thing and how do we do it in a way that could be at scale that that the majority can have access to, which is why I always do ask about lifestyle and things like that, because there are things that can be done now that will slow that process down. And that's more of a statement versus a question, but I do have a question. I just want to kind of, might be philosophical, but just your thoughts on when we think about aging. So this idea of cure aging is more on the, hey, let's stop that breakdown, dysfunction, disease process that will take us out of this body, right? Like let's go ahead. But the real process of aging begins the moment we're conceived. And so it is this constant, like you said, wear and tear process. There's always a building up and breaking down within the body and in the cell from moment one. And so I'm curious, is it being discussed in some of these research communities? Are we really looking at interventions that would be appropriate? Because I think this is one of the moral questions we get to ask, right? At what point do we start where, and we don't know yet. These are more things that probably we don't know, but when do we start certain interventions? And do we start in older chronological age, like a longer life lived, and work back? Do we see the greatest benefit if we were to drop this into somebody in their 60s? And are they going to get the greatest gain? And we'll see it and we'll know it? Or are we looking to really think about, wait, we actually want to start these interventions more in our 20s or our 30s? Do you have any thoughts or research around that, of how we might think about what's coming to market, or when we should be doing interventions, and what's really going to be most in our best interest to enhance longevity?
I get asked that question a lot. My answer is that your mother should be providing you with something to stop the aging process from the day you're conceived. Okay. So it's like even before you're born, you need to be doing something to uh, uh, prevent the wear and tear, or at least the inability to repair wear and tear. And now, of course, FDA might say, pregnant women shouldn't be taking these type of things. So
Mm-hmm.
it doesn't hurt to take it later, but there's no, the sooner you start, the better. Okay. There's none. I mean, yes, if we're like 80 years old and suddenly somebody comes out and discovers something that reverses aging, yeah, that would be something definitely to do, but you probably would have been better to do it before you got to be 80, because
Mm-hmm.
now you have accumulated a lot of mutations, some, in neurons especially, which are very difficult to uh, replace when they get damaged. And by the way, I mentioned I was working on other things. That's one of the things I'm working on, inhibitors I'm working on trying to inhibit the inhibitors that are preventing neurons from being remade when they're destroyed.
Mm.
But that's a whole nother subject. And I'm doing it in collaboration with another company. But
Nice.
it's like the sooner the better. The sooner the better. It's like, as you said, the day you start aging on the day you're conceived. And
Mm-hmm.
um,
Yeah. Yeah. Well, there is a lot to come, and we will be excited for the new research that you're bringing out, for the research you're continuing to work on, to keep our eye on everything, that you've got your fingers in. I want to ask you a personal question as we get ready to wrap up, Bill. I know you mentioned running a little bit ago, and your wife just had a big achievement in the running world as well. So, what is your actual personal longevity protocol? What's Dr. Bill Andrews kind of mapped living as long as possible, as well as possible on a daily basis?
well, yeah, I'm a runner too. I run every single day. In fact, I recently just checked, and I'm something like in 3,500th place in the world right now for the most consecutive days running.
Amazing.
Run over, I haven't missed a day of running since April 9th, 2020, when I forgot to run that day. But so it's like coming up on 5 years without missing a day. But I run at least two miles a day. And then on top of that, I also exercise. But in both cases, I never push it. I never make it so that it can cause stress or inflammation and stuff. So
Mhm.
I run at least an hour a day,
Mhm.
or 2 miles a day. Sometimes I can do that in like 20 minutes. But I also work out with weights and stretching and stuff like that for an hour a day. Um, I also focus on my foods that cause inflammation in
Mhm.
me. I believe that a lot of us have inflammation from foods that we're not even aware of. And so, it's important to get tested for. And I mentioned the ALCAT test before.
Mhm.
It's not an allergy test. It's a food sensitivity test, which is very
Mhm.
different from allergies. Uh, so I would recommend getting those. The, you know, I've been able to, I've done a lot of work myself where I've had uh, phlebotomists be taking blood samples from runners during ultramarathons, before, during, and after ultramarathons, and trying to correlate those with different things. And including myself. I'm an ultramarathon runner, and I've done the blood work myself on myself. And it's like I can actually run 100 miles and not have any inflammatory response whatsoever if I do it right. And that's, enjoy it. Just keep it fun. Enjoy the view. Just don't focus on trying to break some record or something like that.
Yeah.
I mean, the record that my wife just set the world record for the longest lived, and I don't like using the word oldest, longest lived woman to ever complete what's called the World Marathon Challenge, which is she ran 7 marathons on 7 continents in 7 days. She just finished a week ago. She's
Mhm.
still recovering, but she did not go fast. She was just focusing on being the longest lived person to ever do it. And so she took her time, she enjoyed it, and she's tired, but
Yeah.
I don't think she's wrecked her body at all from doing the thing because of the fact that she didn't race to be crossing that finish line by a certain time and
Yeah.
falling on hands and knees, throwing up like a lot of runners do.
Yeah. Yeah.
So that's so so that's one thing that's important. Yeah. And
Mm,
it's not just running, it's endurance exercise. The kayakers just going to have as much benefit to kayaking as a runner is going to have from running.
mhm.
Bicycle, riding, too, uh, all kinds. So it's just endurance exercise. Just keep it fun. Make it an adventure, not something that's a hardship.
Yeah, beautiful. And I think that's one of the best pieces of advice. Make it an adventure, not a hardship. Make it something that is enjoyable to you. So, whatever you decide to do every day, my friends, I hope it brings you joy. Dr. Bill Andrews, today has been a fantastic conversation. Thank you for sharing your personal journey. Thank you for sharing your research, your work, and just helping us to have a greater finger on the pulse of what is coming. We'll be sure to put links in the show notes below about those companies you're involved with and some of the things coming to market that will help us to be able to move the needle a bit on telomerase in our system and slow that rate and pace of biological aging because I'm excited. I believe that we are living in the time that we're definitely going to break that 4 minute mile. We're going to go beyond 125 and I'm sure your work is going to be a part of it. So thank you for your contributions. It's fantastic.
One last comment.
Yeah.
Everybody should go to RAADfest, especially now that it's combined with the Longevity Escape Velocity Conference with Aubrey de Grey. It's like they're not competing with each other, they're joining each other. This is really the best two conferences getting together, and this is going to be the most exciting RAADfest ever this year.
So you heard it. Dr. Bill just took the words right out of my mouth. So if you want to get to meet amazing people just like him, you want to get to sit down and learn from the best, you want to collaborate, you want to be a part of it, please come join us for the 10-year anniversary. Again, links will be in the show notes below of how you can get those early bird tickets. Again, Dr. Bill Andrews, thank you so much for your time and
Thank
for your
you.
knowledge. It's been a pleasure.
Thank you.
Yeah. And to all of you, our guests, continue to be bold, stay curious, and continue to express your greatest states of aliveness. Until next time, I'm Dr. Melissa. Thanks for tuning in. Thank you for joining us on RAADcast, where science and soul ignite the future of radical life extension. Be sure to visit us over at radfest.com. There you can engage with like minds in the super longevity movement and find out about upcoming conferences and events. If you're inspired by today's episode, be sure to subscribe, leave a review, and share it with someone who's ready to live bigger, bolder, and longer. Stay curious, and we'll see you next time on RAADcast.